Good afternoon, everyone, and welcome to Global Hotspots. My name is Christina Zignago, and I am the Senior Program Coordinator for the Wisconsin Alumni Association. Global Hotspots is one of our free alumni learning programs that takes you behind the headlines of today's top news stories with a UW faculty expert. It is sponsored by the Wisconsin Alumni Association, the UW-Madison International Division, and Plato. Now, it is my pleasure to introduce our speaker for this afternoon, Professor Jorge Osorio. Professor Osorio is an associate professor of pathobiological sciences and veterinary medicine. He earned his PhD in animal health and biomedical science from UW-Madison. He is also a major contributor in forming the state-of-the-art research group that uses molecular approaches to study emerging diseases such as Zika, dengue fever, chikungunya, and influenza. Ladies and gentlemen, please join me in welcoming Professor Osorio.
Thank you so much, Christina, for the introduction. Can you hear me okay? Yes, thank you. So it's a pleasure to be here, and it's really nice to see some friendly faces. Good to see Anne, many years without seeing you, and I appreciate you taking the time to come today. I know the weather might be a little, might rain a little bit, but I want to go with you through this world of Zika and emerging arboviruses, so we get to know more about what's going on in the world in this field. So just to start with, I'd like to tell you about what I do in my research, okay? So my group actually studies emerging diseases, emerging viruses. You hear about this in the news, like Zika, hear about dengue. You might not hear that much about chikungunya, but what I like to do is field studies, especially in the winter when the weather is so nasty here. I like to get out of here and go south. So if I know that probably you would like to join me sometime, please go ahead. So I believe that that's important because we have to study diseases in the field. And many days today we travel quite a bit, so actually you'd like to know what's going on, what's happening in some of those areas, and diseases travel quite fast, as we're going to learn from today, as well. At the same time, I use the information from the field to develop vaccines. That's something that I worked with in the past. I work in industry. So I learned there should be a connection between the field work that we do and also developing new vaccines for emerging diseases as well. So in that field, we have worked on a vaccine for dengue. We were able to make a vaccine all the way from the bench to take it all the way to clinical trials, and now it's in phase 3 clinical trials, working in better vaccines for influenza, chikungunya. You're going to hear a little bit about Zika, and so on. And then also we study pathogenesis. We use all these viruses that we collect from the field, and then we have animal models that we use to understand better how these viruses affect the host, with the intention to develop better vaccines and better measures, with the intention of developing antivirals and new ways to control them. So that's basically what I do in the meantime. But what I'd like to do today is basically just go with you through a little bit of what we are doing in Zika and how we're going to study that and control that. Probably you have heard now of Zika in the news. I know that it's probably all over the place in the news, but probably a year ago, you didn't know that much about this, right? It was like, so strange. So this virus was first found in Africa in 1947, okay, in Uganda, in a place called the Zika forest. And it was maintained there for a long time in this cycle between non-human primates and humans. So that's how it came back and forth. And so it stayed there for a long time, and then all of a sudden in 2007, there were some outbreaks that came to the French Polynesia islands, and they were starting to have some other outbreaks there. And then there was another outbreak in 2013 where there were more than 30,000 cases of this disease in the same area. And then you heard the news that it came to the Americas probably around 2013, but we started hearing the news in 2014 about the emergence of Zika in the Americas. So with that, basically this year, because all the news about Zika, the World Health Organization declared a public emergency, which doesn't happen very often, okay? And this was mostly due to the, basically the news of Zika related to microcephaly, okay? See if I can, to microcephaly, okay? And it's expected that about 4 million people could be infected at the end of this year, okay? Which is quite large, okay? We now have Zika transmission in the United States. In the past, we had it in Puerto Rico, but now we have cases in Florida, and we have basically local transmission in the southern part of the United States. Also because of the traveling, so there were many, in all the states, basically, we have cases from traveling people in all the states that go to infected areas. So this is really a major emergency due to the link of microcephaly. And unfortunately, we don't have any vaccine available at this moment. We don't have any antivirals for this that could work. And even the development of a vaccine will take at least, at the best, probably 5 years or so, even if we push it really hard. So it's going to take a long time. And unfortunately, what we have done in the past to control these viruses through mosquito control systems have not worked quite well. We're going to see that as well, so that's an issue as well, okay? Oops. Oh, so sorry. So, but another disease that you don't hear that much in the news here is dengue. And dengue is actually, is considered, more than Zika, the most important mosquito-borne viral disease in the world. That's what we call arboviruses. And this because it really produces quite a few cases. There's about 2 to 5 billion people that live in areas where the disease, is present. So all these regions basically are susceptible to people with dengue. There is about 390 million cases of infection every year. More than 500,000 people get hospitalized with dengue. And this produces what's called the dengue fever, which is also, we call it like the breakbone disease, because basically you feel like your arms and your bones hurt so much. But then it also comes with other forms that are very severe. There's hemorrhagic disease, and basically people even die. So this is a more severe disease that we have. In the Americas, what we had, sorry. So in the Americas, basically, it's a new disease that started about the 1980s, okay? And we didn't have this disease for a while in the Americas, but we had outbreaks started in Cuba in about 1981. There are 4 serotypes of this disease. And then you can see that through the years, we have had outbreaks going on, and every year we see more outbreaks going on and increasing. This slide is from 2010, but if we go to this year, 2016, you see here Brazil with 1 million cases of dengue. This year alone is 1.5 million cases of dengue this year. So it's really a disease that's going up in number of cases every year, okay? And it's affecting many parts of the world. In fact, also in the United States, we have dengue in southern Florida as well, endemic, present there on yearly basis. So it's a major problem. But there's another disease as well that you might have heard of it. It has a strange name that's called chikungunya, okay? And this is a virus that comes from Africa as well. It's from a tribe, the Makonde tribe, which means that bends you up. So what happened in the past, they found people with this posture because they basically couldn't sit straight, And it's basically the long-term effects of chikungunya that affects your joints, as you can see here, and basically is for a long time, for years. This is for years. So the virus was found in 1953. It produces, but we actually have cases in the Americas in the past with the slaves. They came to the States. As early as 1779 in the States as well, okay? Produces a disease very similar to dengue, which is fever, joint pain, this rash in your skin. And This can last for 3 to 5 years, so it's quite chronic and quite severe. Can be also confused with dengue and Zika. In the Americas, we had an outbreak that started in 2013. The virus has been mostly in Africa for a long time. But again, the same thing. Through travelers and through people going through country to country, we had an outbreak that started in the Caribbean islands, okay, in 2013. And now we have a major outbreak going on in the Americas in many places, including the United States, in southern United States. So it's also a major severe disease. There's another one. I want to show you here some pictures of these complications that is going on with chikungunya. Normally, it's just a joint pain, but we actually are seeing very severe disease now with hemorrhages and also some necrosis in some of these patients. So it's also quite severe. There is another disease that you might have heard before. It's called yellow fever. Yellow fever was actually also in the United States. This picture here is from Philadelphia in 1793, where there were major outbreaks of yellow fever, with viruses and everything, mosquito, in many parts of the United States. But the disease continues to spread and to grow. This is earlier this year, we had an outbreak of yellow fever starting in Africa, in Angola. And there was the concern that this disease, because of the traveling that we do, could easily spread around the world really easy. You could take a flight all the way from Luanda all the way to Beijing and basically bring cases around the world. So that's what can happen. Fortunately, what has happened this year with the yellow fever is that there is a vaccine that we can use for yellow fever. So in December 2015, we learned about these cases of yellow fever in Angola, but there was a vaccine shortage of yellow fever. We didn't have enough vaccines, so there was a production of vaccines and started releasing vaccines, and you can see the effect actually. When you actually have good control measure as vaccination, you can actually reduce the number of cases of disease, as you can see here. So it's nice the impact of the vaccine. And now in September, we have few cases of yellow fever. So unfortunately, you can see the impact of the vaccination. Now, why are all these things happening? Okay? Because of the mosquito, as we talk about that. But early in the 1930s, we have plenty of this mosquito called the Aedes aegypti. And then due to the control campaigns that we were doing, we were almost close to eradicate the Aedes aegypti. And in 1970s, very few countries had Aedes aegypti. But then we slowed down our control campaigns, and there were some resistance created with some of these insecticides that are used. And now the picture is difficult to control because many countries have this mosquito vector in many of those countries. Okay? And another issue that also complicates this disease is the presence of another mosquito that we call the Aedes albopictus, or also called the tiger mosquito. It's a mosquito that's very aggressive, that is in many parts of the world, and you can see even in the United States. We have it as, South has Illinois. So we have it in many parts of the States, many parts of the world, Asia and Africa. And this mosquito vector can also be a mosquito for dengue and for chikungunya, and there are still reports, confusing reports, where this is a good mosquito vector for Zika or not. So that's the problem that we have as well. We have these mosquito vectors, okay? There is another disease that I just want to bring to your attention that is just happening. It's called Congo Crimean hemorrhagic fever, which is another strange name, okay? But it's also, it's not transmitted by mosquito, it's transmitted by a tick, okay? But as we always have to watch the news, what's happening, what's new happening, okay? So I found this report of, in New York Times, okay? Basically of outbreaks in Spain, happening as we speak right now, where there is about 200 people that have been sick with this disease, and it has about 40% mortality. So we have these viruses, emerging viruses, in many parts of the world, and we have to stay vigilant. So that's part of the work that we need to do as well. Okay? So what are these viruses? So I don't want to give you a virology class, but just generally just tell you they are very similar, okay? That's what it is. They are very similar viruses. Yellow fever, dengue, and Zika are from the same family, okay? They're the same group of viruses, okay? So they look very similar to each other. There is another virus there that is in Japan called Japanese encephalitis virus that is also very similar. We heard in the past of West now, so it's also something that's from the same family. And we have other members, St. Louis encephalitis in the United States, and there's other ones transmitted by ticks, tick-borne encephalitis. So they look very similar. The chikungunya virus is a little different, but again, it's transmitted by mosquitoes. It's a different family, and that family we have the alpha viruses, we have what we call the equine encephalitis viruses, Eastern, Western, and Venezuelan equine encephalitis. But overall, they tend to have something similar in the structure, but the genome is a little different. But we need to know this because we need to make vaccines and study these viruses. So that's what we use. So why are we having all these new viruses spreading? So obviously, the first thing that we have to see is the flights, the number of flights that we have per day. I Googled how many flights we have a day, and there's over 1,000 number of flights a day that we do around the world. So amazingly, you can be in any of these places, and the next 12 hours, will still have fever or so, you can be back in the States or somewhere else. So that's what's contributing to a lot of the transmission that we're seeing with these pathogens. Okay? But in addition to that, we have other problems as well. We don't have the immune system that can protect us against some of these pathogens, right? We don't have the antibodies. We have not been exposed to some of these viruses, so they come here and they find a totally naive population that basically the virus can spread easily from people to people. Some of these viruses are also very clever. They tend to change a little bit, okay? They have a way to mutate and adapt to new conditions and new hosts. The chikungunya virus learned to make a small change. It was maintained in Africa, but it learned to make a small change, and from that infected a new species of mosquito called the Aedes albopictus, the one that I mentioned, the tiger mosquito, and with that it spread much easier around the world, and it went to Asia and to Europe. So the viruses make some changes. And also the environment. We have a globalization issue. The cities are bigger and bigger every time, we have that. And then we also have the climate change. So we have mosquitoes in areas where they shouldn't be. Normally we talk about mosquitoes that couldn't be above 1,500 meters. Now we find these mosquitoes, 2,200 meters, for example. So there's new areas that are exposed to these mosquitoes as well. And you can see that the climate, some people might say that the climate, winter here are tough, but you can see that actually they're softening a little bit. The climate has affected that as well. Okay? So what I'd like to tell you is what we are doing in terms of studying these viruses, okay, with the work that we do. First, as part of educating the community, I'm a member of what's called the Global Task Force. Those are basically our group of individuals around the world where if there is an outbreak like this happening, or we need to talk to the community or the media, we do that, okay? And also if someone needs some support, some help, or reagents, or there is a strange disease happening, we are part of this group of this community. So we're very fortunate in Madison that we have a very nice group of investigators that are pretty good there in terms of collaborating with the scientific community. We have field sites, many places around the world, mostly in the Americas, where we actually study some of these viruses. So do some studies in Mexico, in Colombia, Ecuador, Brazil, and Argentina. And because of this field work, we were able to first characterize the Zika infection when it was happening late last year. So we were able to do that. And then we actually do what we call cohort studies, okay, to study what's happening. And so we received a call from NIH, National Institute of Health, to be part of international collaboration to study the effects of this infection in pregnant women. So we are doing that study, and I'll mention that a little bit. We're also developing animal models. That's what we are doing. Non-human primates, you probably heard on the news the work being done in Madison with the non-human primate studies, which are basically leading the world right now in terms of what we learn about Zika infections, because obviously it's not very ethical to do these studies in humans. So we have to find animal models to do that. So we use non-human primates, and we use mouse models to do that. Okay. So, well, we're also studying some ways to control this. There's a bacteria that I'll let you know more about that. It's called Wolbachia. And that we are using that bacteria as a control method for these diseases as well. And we are doing some development of vaccines, trying to find a vaccine, particularly for Zika. We have developed vaccines for dengue and chikungunya, but we're now trying to develop a vaccine for Zika. Okay? So this is what I plan to tell you today, what we are trying to do. So in terms of how do we do these field sites, we are basically here in Madison, but we have all these sites around the world in the Americas, in Ecuador, Colombia, and Argentina. And in Colombia, we have sites that are either on the northern part of Colombia, okay, Santa Marta or Sincelejo, Medellin, which is a big city, about 2.5 million, where we study in urban areas, okay, or at the southern part of Colombia by the Amazon River, which is called Leticia, where the Amazon River goes by there. And that gives us a chance to study these diseases in the environment where basically they are being transmitted frequently, or just to observe what new pathogens are coming out of there. So that's what happened when last year we were observing some patients that came with a febrile illness that it looked like dengue, but it was not dengue. So we studied that disease right away in that town called Sincelejo, and we were able to detect the Zika virus. That was in October or November last year, okay? So, sorry about that, someone is looking for me, I guess. So what are we doing in terms of looking for how to make a better vaccine for Zika? So we are using non-human primate studies here in Madison. And the first thing that we learned is that monkeys are susceptible to Zika infection. That's something that we learned, okay? The second thing that we learned is that when actually we infect pregnant monkeys, they are infected for a very long time, and they can actually shed virus for a long time, which is very, it's something that we need to learn this first from a model, and then go and test in people in infected areas to see if this is really true. And this is important because if the mother is infected for a long time, that gives, that creates an issue because exposing the fetus for a long time as well. So that's something that for us is very important to learn as well. And then we also did some studies towards a vaccine development, in the sense that we can infect an animal, okay? And what you look here is the amount of virus in the blood, okay? And then later on, about 2 months later, we infect them again, and we see the animals do not develop that, what we call viremia, virus in blood. So that tells you that we actually could develop a vaccine that could protect against Zika, because the animals do not show this peak that you see here. You don't see it here again. So that's something for us good to know towards the development of a vaccine for Zika. So we also have developed a mouse model to study the infection, because the non-human primates, they don't die. They just develop, the virus is in the system, but they don't get sick. But we need to develop a vaccine, we need a model that shows mortality. So we have an animal models in the lab where we actually can infect mice, and we can see that actually they get sick and they die. These are the days, they die within 8 days post-infection, so they die really fast, okay? And if we look, they're different ages, if they are young or adults or so, they all will die in that case. And we look here, they lose weight. The animals basically all lose weight very quick versus the control animals, they stay very healthy. So that's a good model for us to understand and to learn more about the disease itself. So with that, we look at the tissues, where the virus goes in the tissues. We have characterized the virus in the brain, in the muscles. We have virus in all the tissues in the animal. We see where it goes, and we see the amount of virus that we have in the blood as well. So this is work that we have done to characterize the infection in an animal model. We do that with the idea of developing a vaccine. So what we did in our group is actually with the goal to develop a vaccine. We we had the virus in our lab, and we do molecular biology to express what is basically the outside of the virus. That's what the immune system needs to see, what is outside of the virus. And then we make this nice little thing, which is called VLPs, okay? Which is right here. Basically, what you see here is how the virus actually looks like, okay? So it is like the shell of the virus, but without the ability to make you sick. So if we take that and we put that into a mouse, we actually see that and then give them the nasty virus, we see that actually the mice all live. They do well and they live, versus the control group that they all die really, really quick. So that's the first steps that we're doing towards developing a vaccine for Zika. And we look at the weight for these animals, and we see that these animals, basically, they are vaccinated, they don't lose any weight. Compared to the control groups, they all get sick and die. So that's the efforts that we're doing from here. Obviously, there's still a lot of work that needs to be done to take it from here to another model, and eventually it's got to go to clinical trials, but it will take some time to do that. So I mentioned that we're doing some studies to understand what's going on in the field. So this is a nice collaboration between 5 different countries, Brazil, Colombia, Nicaragua, Puerto Rico, Guatemala, to study what's happening in the field, because the data from Brazil, when we see all this data of microcephaly, it really is very strange. There are more than 4,000 cases of microcephaly in Brazil, but in some of the other countries, we are not seeing the same number of cases. So we wonder what's happening, and the best way to know that is by comparing data across the countries and doing collaboration. So we have that collaboration where we are creating a large cohort of more than 10,000 pregnant women, and that we are going to see what the outcome of the infection is. So in that study, we plan to enroll women in the first trimester of pregnancy, and then we do a physical exam, and we will collect ultrasound information, but at the same time doing health questions and collecting samples of blood and urine through this whole study, and then looking later on and see the effect on the newborn. We will follow the newborn for at least 1 year after they're born to see if there is any effect of the infection in them, okay? So basically this study is just starting, ongoing, and we'll have data. It's gonna take a while, but at least it's a start to really understand what's going on in the field. All right? So now I'd like to tell you a little story about something that's called Wolbachia, okay? And I forgot to say that I need an audio here, but let's see if this works.
The mosquito known as Aedes aegypti originated in Africa. Over the last 400 years, it is spread
Is that too
across
loud?
all tropical regions of the world, transmitting viruses to people. We are working on to stop this disease from happening in dengue.
I'm sorry, I just trying to figure out the sound. Oops, sorry. Let's see.
The mosquito known as Aedes aegypti
Do better?
originated in Africa. Over the last 400 years, it is spread across all tropical regions of the world, transmitting viruses to people. We are working on to stop this mosquito from transmitting dengue. Almond produces a naturally occurring bacteria called Wolbachia that lives inside insect cells and are passed from one generation to the next through the insect egg. Wolbachia are found naturally in up to 60% of all the living species of insects. Insects that include fruit flies, moths, dragonflies, and butterflies. The secret to the success of Wolbachia is how it imitates the reproduction of the insect it lives in, so to give itself an advantage. It works like this. If a male insect has Wolbachia and makes it a female pregnant, then the egg she lays works. If the female has Wolbachia and the male doesn't, she will lay a normal number of eggs. They will all hatch, and all offspring will carry Wolbachia. When two insects that both carry Wolbachia mate, the eggs will hatch, and all offspring will carry Wolbachia. Over a few generations, the number of individuals carrying Wolbachia increases rapidly until all the insects within a population have the bacteria. We want to would use Wolbachia as a way to stop dengue transmission. To do this, we first set a Wolbachia into the mosquito to let it grow and be passed between generations. Using microscopic needles, we took Wolbachia from a fruit fly and injected it directly into young Aedes aegypti eggs. It took us thousands and thousands of attempts, but finally we were successful. We then took the mosquitoes that contained Wolbachia and infected them with dengue and found something really exciting. The virus didn't grow well in mosquitoes, and if it can't grow, it can't pass between people. Our next step was to build large enclosed outdoor cages in northern Australia that were filled with little mosquitoes. We then introduced mosquitoes containing Wolbachia to see if it could spread into the cage population, and it did. We now know to test the method where dengue is found, but before we did this, we needed the government to examine the safety of our method and approve our trial. Then we needed to gain the support and approval of communities to release mosquitoes containing Wolbachia. With these approvals, we began our first open field trial in Australia in 2011. Wolbachia mosquitoes were released once a week for 10 weeks, and within a few months, close to 170 mosquitoes had Wolbachia. And years later, they still do. Ongoing trials in Australia and many endemic countries are now determining the best way to deploy Wolbachia across whole cities. It is our hope that with continued success, the Wolbachia control measures protect the health of 2.5 billion people living in heavy transmission areas.
Okay, so that's what we are working on. It's a bacteria called Wolbachia. Okay, and I'll welcome you to visit that website if you'd like to learn more about this. Okay. So to summarize, basically, it's a naturally occurring bacteria. It's actually 60% of the insects have Wolbachia. Butterflies have Wolbachia, fruit flies have it, so it's naturally occurring bacteria, okay? So it's pretty safe. It doesn't have any effects in humans, okay? And it can only be transmitted through the eggs of the mosquito. So that's where we find it. And again, as the cartoon explained, basically the movie, we have it through the breeding of either females infected with Wolbachia, with males infected with Wolbachia, or females along with the wild-type mosquitoes. They all have Wolbachia. So what we like to do is eventually replace the population of wild-type mosquitoes with these mosquitoes that actually have Wolbachia. Okay, that's what we like to do. Now you wonder why we like to do that. But there is data that actually this is a very sustainable system. That if you do this for a short period of time, you release these mosquitoes for a short period of time, the population is maintained for about years and years to 90–95%. And the indications from studies in Australia, these are 2 communities in Australia, is in those areas they have not seen dengue transmission. So that's why we like to do that. We have data with dengue showing that this is too technical, but this is basically to show you that you can actually reduce the percentage of mosquito infected that transmit dengue if you use this system. So these are 4 different serotypes of dengue, and you can show that you can reduce from 70 to 80% of the transmission on those mosquitoes. So it works quite well. We also have done that in our lab here for Zika, and have shown that basically when we have these mosquitoes infected with Wolbachia, or the wild-type mosquitoes from Colombia, and we give them virus, we give them in a mouse, we give them the virus. And basically what you have to look is this data here, is the number of mosquitoes that are transmitting Zika. And the wild-type mosquitoes, we have about 60% of the mosquitoes that transmit Zika, but the mosquitoes infected with Wolbachia, we have 0 transmission. So it works quite well. So it basically, it blocks the transmission in the mosquito. So we have done something similar for chikungunya and actually found very similar results. That the mosquito wild type transmit chikungunya for quite a bit, okay? But the mosquito with Zika basically does not transmit. So this is the approach that we would like to try in the field to reduce the transmission of these diseases. So with that, we are working with a group called the Eliminate Dengue program that strike with different sites around the world to do this. And they have, this group started in Australia. This was discovered at Melbourne, in University of Monash, but they have set up some field sites around the world, in Indonesia, in Yogyakarta, Vietnam, in Natran Island, and in the Americas, we have a site in Brazil and one in Colombia, okay? So what I'd like to show you is the work that we are doing in Colombia to do this. Okay? So the work in Colombia is done in Medellin, collaboration with this team, there's Dr. Ivan Vélez and his group there, okay, to study the biological control of this disease, okay? And the city is about 2.5 million, okay, which is shown here. We are working, the city is in the central part of Colombia. We are working in the northern part of the city in a community called Barrio Paris, Neighborhood Paris, where it has about 44,000 people living in that area, okay? So this gives you an idea of the location and the place where we do this work. Basically, as you can see, it's areas of low income, you know. You see a lot of people in high density, and that's actually where most of these viruses are mostly transmitted, in areas where there's actually a very high density, a lot of population, okay? And you can see there is hillsides, basically it's on a valley, in a hillside, so it actually makes it difficult to work there as well, okay? And also shows you that because these are coming from, people coming from many parts of the country, there is this new settlements. This is how some of those neighborhoods start. They basically build a cardboard house, and then from there on, they go on and on and on. So this is some of the challenges that we have to work there. We work with the community. That's the #1 thing that we do. We have to explain the projects to the community. And one of the successes of the project is that we were able to engage with some nonprofit organizations, community foundation, to really do this work, okay? So we do scientific talks with the scientific community, but also with many different people in the area as well. We go to the schools, and we teach in the schools about mosquito diseases, about Wolbachia, what it is. And we get a lot of support from volunteers, which basically have endorsed the program and have become trained on what we are trying to do. And basically, we're hired through the program to do the community work, because everybody knows them in the community. So that has been really good for us, okay? We also have created fever clinics, and that's also important because we need to know if this has an impact or not in the community in the disease. So we created fever clinics where we have medical doctors, and anyone that has fever gets treatment. We come, we see what it is. We have improved the knowledge about dengue and Zika and chikungunya in this area, knowing how many more cases they have. And also this helps us because now we can use GIS, you know, to reference the locations of the cases of those patients, where they are getting sick. And we know the different serotypes that they're getting sick. And so this is the map of the community. This is the area where we are planning to release. And here you can see that there are actually many cases that are happening outside of the community area. So that's important for us to know where the cases are coming from for the future, okay? And with the volunteers, with the community, we do the releases of the mosquitoes. We produce the mosquitoes in Colombia, and they go by certain routes, and they release the mosquitoes with the support of the local community. So that's also very important because we are very transparent of what we are trying to do. To do all this, we have to have consent from the whole community, 95% consent. And they all actually accepted to participate in the study. So, so with that, we also learn, so we do the releases for a period of time, you know, about 8 weeks, and then we measure how many mosquitoes there in that neighborhood have Wolbachia. And we collect all that data through the year. So this movie here, just to show you through the year how that population has Wolbachia. So we actually have learned basically that if you go through the year, the Wolbachia has established itself there in that population. and basically is maintained. It's maintained. So with this in mind, the community has asked us if we could continue with the study and we could go through the next neighborhood or the other neighborhoods where you remember you saw those cases of dengue that we had there. So that's now our next intention is to actually do this in a much bigger area. So the next step, it will be a challenge to do a city of 2.5 million, which that's what we like to do. If you can imagine, we're going to produce more than 800,000 mosquitoes a week and release 800,000 mosquitoes per week. But that's a challenge. But we feel we have the support of the community, we have the support of the government to do this, and we will start in a small scale. This is where we have started now, okay? And we will start spreading by different phases, and we will continue growing through the whole area, okay? So the plan is to do this for the next year, okay? And with the goal to really replace the mosquito population with Wolbachia mosquitoes. Here's a movie, basically what we like to do. You might get this in the mail too. So we might actually put them on a box and mail them in an envelope, and you can just take them home. And the mosquitoes basically have eggs. Those are eggs that are in that little red strip there. So you put that with water, and the mosquitoes basically will start from there. So you can save your Chinese boxes, for this project later, okay? Okay. That's it. So with that in mind, we actually have worked with some folks that do what's called mathematical modeling. They take the numbers and see, with the information that we have from this area, what happened in the future if we release Wolbachia and replace the mosquito population with Wolbachia. So what you see here on this map is basically going year by year, the epidemics that we're going to have on dengue in the area. And because we have pretty good data on the whole region, we know when the outbreak's happening, okay? How often, so you can see that there's going to be peaks happening every 5 years or so. And there's going to be a new peak coming up that is going to be very large, probably in 3 or 4 years. But as we are progressing and replacing the population, you're going to see that some of these peaks actually start going, reducing a little bit, okay? And then you start seeing less intensity in the future of those peaks for dengue. And eventually, the goal is that basically in about 20 years or so, we start seeing this. We start seeing just basically very few cases, okay? And eventually gets to a point where you see no more cases. So that would be our goal to do that there, okay? So this is a model, but I think basically we base that information on historical data that we have of the disease in the area. So that's what we'd like to do, okay? So just in summary, we study dengue, Zika, and chikungunya here. We use Wolbachia, which is a bacterial strain, has a biological control approach that we'd like to do that, okay? We are planning to do we are doing field studies in parts of the world. In Colombia, we are doing some of that, we plan to do a large deployment of 2.5 million, okay? We're also working on a vaccine development for Zika, and we are planning to do some field epidemiological studies to study Zika infection in pregnant women to understand more about Zika. So with that, I just would like to finish thanking for your attention. Also thanking the team here at the University of Wisconsin, but also the team in many parts where I work in Colombia, also in Monash University, and the community where I do some of this work. Thank you. So be happy to entertain some questions if you have some. My microphone will come.
Is Wisconsin the only place that this type of research is being done in the United States?
It is. It is really. Actually, we lead in that. In fact, we were prepared when we heard of the Zika epidemic. We were very prepared, and we were very fortunate to have a really nice team of faculty and very good interaction between us, because right away we basically jump in, into this, and the National Institute of Health recognized that effort, and we are leading on this area right now.
Am I wrong to assume that you were saying that eradication of the mosquito, as they are currently doing around the Miami area, that you do not see that as a viable long-term solution?
Well, you read my line, so, but it's true, because what I see difficult there is how you sustain those programs and the cost of sustaining those programs. And it could work for some communities, but we're also talking about, low-income countries where they have to put many of the resources to fight these diseases. So if their budget is going to go to every year release new mosquitoes, like what you're talking, the most sterile mosquitoes or so on, that's going to take a lot of their budget. So it can work for some communities, but has a large picture where we have some low-income countries that are trying to find a solution that will be difficult to do. Okay.
Yes, I noticed that in your first slide you were doing some research also on white nose syndrome.
Yes.
Could you say a little about that?
I
That's
could say
serious
that.
in Wisconsin.
It is a very serious problem in Wisconsin, so I'm very fortunate that we have very nice collaborations with the National Wildlife Health Center, the DNR, and it's a major problem that we have. So our intention is to create a new vaccine for white nose syndrome. Okay, we have some very encouraging data, okay, on that as well, and we are actually about to start a study in this winter, because the problem with this is that the bats go into hibernation. Okay, and what it seems to happen is that they actually wake up earlier from hibernation and they die. That's what the white nose syndrome does to them. So to plan those studies is really difficult because you have to have hibernation chambers, and you actually have to plan them in advance because you only get bats certain times a year. So we actually, but we are very fortunate. I think we are doing a study this fall to actually see if this vaccine that we have works. Okay, and if so, you'll hear next year. Okay. How do you vaccinate bats? That's a good question. Very carefully. That's the problem. So what we do is that we work with viruses, viral vectors. Okay? So there are viruses that can infect by mucosal route. And in fact, we are doing that in wildlife. We spray baits into the field, and the animals consume the baits and they get vaccinated. So obviously with bats it's a little more tricky, but what happens with bats is that they tend to be very social. They live in colonies. So they are in contact with each other, if you see that. So that's what is being used to control them for rabies, for example, because they put a paste with warfarin, and they lick each other. And they consume that, and that's how they control themselves. So the same thing we can do with the vaccine. We have done studies that we put the vaccine in their fur, and they lick it, and they get vaccinated. So we have done that. So that's what's going to happen.
For the questions, please wait till we give you the mic just so that everyone can hear you in the room.
Okay.
Thanks.
So I heard about a similar release program in California of
Correct.
mosquitoes, and I know they were only releasing the male
That's
mosquito.
true.
So what impact does your release of the eggs have on the overall mosquito population?
That's a good question. So there are different, because of time I didn't explain this, there are different strategies. We're talking about, what I explained here is replacement of the mosquito population. What the approach is in Florida and in California is called suppression of the mosquito population. They are trying to basically reduce the mosquito population. And the same thing, that system has to continue all the time. You have to continually release, because those males basically are going to breed with the wild type mosquitoes, okay? And they have to replace the mosquitoes. So it's constantly suppression. So that's the difficulty that I see those programs, because basically it's difficult to sustain for the long term. You have to release mosquitoes all the time. So that's the difference.
But does yours increase
Ours increase the mosquito population sustained, yes. But the mosquitoes that are there are not able to transmit. And also, we also feel that in our environment, mosquitoes are needed for something, okay? Bats live out of, there's a lot of bats that live out of the mosquito population as well. So we don't like to play with, changing the equation between one species and the other. So we feel that it's better to control the diseases by replacing the mosquito population instead of totally reducing it, suppressing it. Microphone.
So the mosquitoes that carry these viruses, we notice them in Florida
Yeah.
and in warmer places. Do you expect them eventually to move north, or is it
We will be dead by then, but
What would you say? What?
I said we might be dead by then, but the climate is changing, okay?
Yes.
Obviously, and they're moving north, okay?
Right.
Because the conditions are changing in our environment that before it was in Florida, but now they're now in Texas as well. You find them, and you might find them pretty soon in Georgia,
Right.
or so.
But it's not like they're on their
No,
way here.
no. Now, what could happen,
Yes.
which I don't want to scare you, is that if the viruses learn to adapt to a new species of mosquito, which we have here. So if they learn to adapt to the Aedes albopictus, the tiger mosquito,
Yeah.
we have that in Illinois, we have that in Chicago, and we could have it very soon in southern parts of the Wisconsin part as well. So that's the concern that we should have.
But so far they don't, but that's...
So far, they have not changed, they have not adapted to that, but that doesn't mean that they might not try in the
Right.
future.
Okay.
So that's what will happen. Here.
Is the NIH your only source of support for this?
Well, we had to try multiple sources, okay? Donors, I should put my hat out there and see if I can collect any money. But what we do at the university is that we try multiple sources. Obviously NIH is the main one, but it's very competitive, okay? But you compete for excellence too, and that actually creates good spirit, creates good science at the same time, okay? So we try that. The Gates Foundation is very good on this. In fact, this whole Wolbachia project is funded through the Gates Foundation, okay? The Wellcome Trust in the UK, I don't know if you're familiar with that one as well. So USAID as well. So there is funds through the government that are being put to this, okay? The Zika emergency actually is very interesting because we have been talking about dengue for a long time, but never had the impact on public opinion that Zika has had. Because when you see those pictures of those babies, really that makes a big difference, okay? That's really what has had a major impact this year to see this. And it has, unfortunately, we are in a election year and politics got through funding and everything. So unfortunately, NIH right now, the Congress, I have to say this, Congress has not released the funding for this year for these programs. So we're still working with pennies, unfortunately, because we are caught in a political year. So that's unfortunate. Any more questions? Yeah.
Is there any worry that Zika or dengue would mutate to the point that Wolbachia would not
Be effective.
stop the transmission and in fact it might strengthen it?
That's a great question. I was expecting that actually, So what will happen? I think what will happen is that if we do nothing, okay, we will see these peaks every year, okay, that we see of we do nothing, okay, continue having the peaks. Now, there is the work of a vaccine going on. That's something I want to tell you as well. That is not very effective, unfortunately, but it's something. And if you have the vaccine, which is maybe 50% efficacy, and the Zika and the Wolbachia works about 70%, you can have some effect there, okay? I'm sorry, this is, if the vaccine is a 50% efficacy, and you vaccinate about 70% of the people, you see some effect. If you do the Wolbachia alone, you see an effect for about 30 to 40 years, okay? Until what you just said, that could be that the Wolbachia or the mosquito, they mutate and they learn how to change each other. By then, you might have time to come up with a new strategy, another strain of Wolbachia, that you can do that. But then if you actually combine the vaccine and the Wolbachia, you can have an impact for a long time, for over 40 or 50 years. So that's why I also see a combined strategy of doing vaccine work along with control methods as well. Okay. So thank you for the question. Any more questions? Thank you.
Right now, Zika is more just flu-like symptoms, right? It's not a dangerous disease in itself.
Yeah. So
is it possible that it could mutate into something more like dengue?
More severe. Good question. So I didn't mention this, that 70% of the cases of Zika, you don't feel anything, are asymptomatic. Basically, it's like even nothing. The rest is mostly a flu-like disease. In very few cases, you have this other rash and everything else. It's possible, and I don't want to scare you, what we call enhancement, because these viruses are very close to each other, dengue and Zika. So what we don't know yet what's going to happen in areas where we have dengue, okay? And then people get infected with Zika, are we're going to see what we call enhancement of disease, which is a disease that is more severe, okay? There are some laboratory studies that are going on right now that it might indicate that that might be the case. So that's why also it's good for us to have some field studies to monitor if that's actually the case. So that's important.
I was wondering too, with the pregnant women, is there any studies that are done about when they had when they got the disease?
And
I mean, late in pregnancy, I can't see where it would affect the baby that way.
Yeah.
But maybe I'm wrong.
No, there are some studies that are already happening. Which trimester of pregnancy is the most, they could have most devastating effect on the fetus, and that seems to be on the first trimester of pregnancy, and that's why our studies now are focusing on that particular time, because that's when a lot of development is happening there. So that's when it could have a major impact. Yeah, good. One more.
How does malaria differ from the diseases you spoke about today, since, I mean, both types are carried by mosquitoes?
Yeah, it's quite different. Malaria is a parasite, it's a big bug, okay? So it's huge. So that's a very difficult, obviously a very difficult beast to deal with. We have no vaccine for malaria, okay? There is some work being done, a vaccine in Africa, which is about 20 or 30% efficacy, a little bit. And it changes quite a bit. What happens with malaria is that once what's called switches the antigen. So once it has one presentation and then later on changes quite a bit, mutates quite a bit. So it's quite challenging to have a vaccine for malaria. Very difficult. Okay? Well, thank you so much.